What a fascinating sight! A gigantic, shimmering green carpet of algae stretches across the German Bight, so big that it can only be seen from up here. During the annual spring bloom, tiny algae multiply so massively that they seemingly become one giant organism. In spring, the sun shines longer and stronger each day, and after the winter, there are plenty of nutrients in the water. In these conditions, microalgae in the water, so-called phytoplankton, can grow particularly well. A natural spectacle occurs, an algal bloom. Algal blooms are important because algae form the basis of the food web in the sea. Thus, the existence of all ocean life depends on them. And also our life is strongly dependent on the tiny algae. They produce about half of the oxygen we breathe and take up a lot of carbon dioxide from the atmosphere. Algal blooms are short-lived. The algae die when they run out of nutrients. Then the organic material they contain oozes into the seawater as sugar-containing compounds. Immediately, bacteria start gobbling it up. An algal bloom is typically followed by a bacterial bloom. The bacteria break down the remains of the algae and return the nutrients to the sea. And so other marine life, such as fish, also benefit from algae production. Not all algae components are eaten by bacteria. The remains sink through the water and to the seabed. There, plenty of other microorganisms are already waiting to eat them. Around 500 million bacteria live in just 1 teaspoon of sediment. Only what they leave uneaten of the algae then sinks deeper into the sediment and can be buried there for a long time. Researchers at the Max Planck Institute for Marine Microbiology want to understand exactly how all this is connected. Therefore, off the island of Helgoland, they are investigating which marine bacteria react to blooming microalgae and how. In order to do this, they use modern methods allowing them to take a closer look at what's going on. Bacteria all look pretty much the same at first glance, so the researchers use their genetic material to tell them apart. They enlighten the darkness with a method called FISH, short for fluorescence in situ hybridization, in which the ribosomal RNA is stained with fluorescent dyes. Special microscopes also allow researchers to look through single cells and see what is happening inside them. The Max Planck researchers were thus able to see that some bacteria sometimes take up long sugar chains which are actually too big to eat and hoard them between their 2 cell walls in order to then break them up at their leisure. That way, no one can take these sugars away from them. Another method called flow cytometry enables them to analyze thousands of cells in a second. Generally, the more common a species is, the more important it is to the ecosystem. But sometimes, you need to see how active the bacteria are and how fast they multiply. If bacteria divide often and are still rare, this indicates that they are a popular victim of larger predators or viruses. Metagenomics is a method of analyzing the DNA of microorganisms directly in the seawater samples, even those that cannot be grown or stained in the lab. The researchers found that algae in spring blooms vary quite a bit from year to year. However, the bacteria don't seem to care. The same groups come to feast on the algae every year. Looking at the sea, whether from the shore, aboard a ship, or from a satellite image of the Earth, it appears as one vast, quite featureless habitat. But the closer you look, the clearer you see. The oceans are highly complex, a finely structured network of life. Even the smallest ocean inhabitants are of utmost importance. Only if we understand them better, we can make short and long-term predictions about this ecosystem and find better ways to take care of it.